At the University of Freiburg, Germany, researchers from the Institute of Biochemistry and Molecular Biology, the Biocenter for Biological Signaling Studies, and the Spearman Graduate School of Biology and Medicine teamed up to study the biogenesis of mitochondria. Mitochondria are the powerhouses of eukaryotic cells. They possess two membranes. The inner membrane harbors the respiratory chain, which converts the energy from food to power reactions in our body. Mitochondria contain more than 1,000 different proteins. Most of the proteins are synthesized as precursors on cytosolic ribosomes.
The precursor proteins are imported into mitochondria by the translocase of the outer membrane called TOM complex. The precursors contain signals which are recognized by TOM receptors, followed by translocation through the TOM40 channel. TOM40 is a beta-Burl membrane protein containing multiple beta strands. Beta-Burl proteins are present in the outer membranes of mitochondria and chloroplasts. According to the endosymbiotic theory, both organelles have been derived from gram-negative bacteria which also contain beta-Burl proteins. Beta-Burl proteins are essential for metabolite translocation, signaling and protein transport. The TOM complex is the main protein entry gate of mitochondria and is used by 4 different import pathways. Precursors with amino-terminal pre-sequences are transferred to the pre-sequence translocase of the inner membrane. Hydrophobic metabolite carriers are inserted into the inner membrane by the carrier translocase. Cysteine-rich proteins are imported by the mitochondrial intermembrane space assembly machinery MIA.
Beta-Barrel precursors are imported by the TOM machinery but need a further protein complex for insertion into the outer membrane. We term this complex the sorting and assembly machinery or SAM complex. After translocation through the TOM40 channel, beta-Barrel precursors bind to chaperones in the intermembrane space and are transferred to SAM. It has been unknown if TOM and SAM cooperate and where the precursor proteins are folded to the mature beta-Barrel proteins.
We established a position-specific assay to study beta-Barrel formation in intact mitochondria. We inserted cysteine residues at defined positions to monitor disulfide formation in outer membrane. Beta-Barrel folding occurred directly at the SAM complex. This non-quantitative assay enabled us to screen for Easter mutants affected in beta-Barrel folding, and unexpectedly, the central import receptor TOM22 was required for efficient beta-Barrel folding.
How can a TOM receptor affect beta-Barrel formation which occurs at the SAM complex? So far, it had been assumed that TOM and SAM are two independent protein machineries. We performed several assays to study if TOM and SAM complexes interact with each other. Pull-down experiments with lysed yeast cells, chemical cross-linking, and affinity purification followed by quantitative mass spectrometry. All these results indeed show that TOM and SAM interact with each other.
We were able to isolate the TOM-SAM supercomplex via native gel electrophoresis. The supercomplex consists of one TOM and one SAM complex. We further screened for yeast mutants which affect the interaction between the two complexes. It was found that the receptor domain of TOM22 is crucial for the formation of the supercomplex.
What are the implications for the evolution of mitochondria? In gram-negative bacteria, beta-Ball proteins are synthesized in the cytosol, are transported across the inner membrane and inserted into the outer membrane by the beta-Ball assembly machinery BEM. The central subunits of BEM and SEM, BEM-A and SEM50, have been conserved from bacteria to humans, suggesting a similar mechanism for beta-Ball folding. However, the sites of precursor synthesis are different as mitochondrial precursors are synthesized outside the organelle. The TOM-SEM supercomplex couples protein import and protein export at the mitochondrial outer membrane and thus promotes biogenesis of beta-Ball proteins by substrate channeling. Factors on both sides of the membrane, the import receptor TOM22 and the intermembrane space chaperones, are required for efficient transfer of the precursors. Beta-Ball formation may thus drive protein import, since beta-strand association will pull the precursor through the TOM complex.